Mumbai, and he has been the consultant at Leelavati Hospital and Bridge Candy Hospital. Sir has been the guidance to put together such a fantastic program with esteemed faculty and speakers. And today's topic is about childhood acute lymphoblastic leukemia, how to manage in 2022. Without a further ado, now I hand over meeting proceeding to Dr. M.B. Agarwal, sir. And thank you so much, sir, for your guidance. And over to you.
Thank you, Dr. Arvind. So, good evening, friends and colleagues. Today, our guest speaker is Dr. David Tichy from Philadelphia. He will be lecturing on childhood acute lymphoblastic leukemia, how to manage in 2022. This webinar is brought to you by Mumbai Hematology Group. It is supported by MCure, managed by Mice Ideas. I thank Mr. Keyur Soni, Dr. Arvind, and Manisha Sonavdekar and the team MCure for supporting our academic activities, Rajesh Kalpesh and team Mice Ideas for managing them, Executive Committee of MHG, our guest speaker Dr. David Tichy from Philadelphia, all our discussants who are eminent hematologists and hemato-oncologists, you participants for sparing your Saturday evening. Tomorrow morning, we have our weekend webinar at 11:30 a.m. and this is on rare hematological disorders. We are going to discuss 3 diseases and the faculties from Manipal Hospital, Bangalore. Discussants today include eminent hematologist oncologists from Mumbai, London, Nepal, Chandigarh, Delhi, Mangaluru, Aurangabad, and Belgavi. Dr. S.D. Banavli, Director Academics, Professor, Department of Medical and Pediatric Oncology at the prestigious Tata Memorial Center, Professor, Homi Bhabha National Institute, Mumbai. Dr. Archana Swami, Consultant Pediatric Oncologist, Hematologist at Bijewada Hospital for Children, Parel, Mumbai. Dr. Chetan Dhamne, Associate Professor, Pediatric Oncology, Tata, Mumbai. Dr. Akanksha Chichra, Assistant Professor, Pediatric Oncology and BMT, ACTRAC, Tata Memorial Hospital, Mumbai. Dr. Ruchira Misra, Senior Consultant, Pediatric Hematology Oncology and BMT Unit at SRCC Children Hospital, Mumbai. Dr. Preeti Mehta, Pediatric Hematologist Oncologist from Sir H.N. Reliance Foundation Hospital, Mumbai. Then we have Dr. Khushnuma from Great Ormond Street Hospital, London. Dr. Richu, she's Consultant Pediatric Hematologist Oncologist from Purbanchal Cancer Hospital, BNC Medical College, Zappa, Nepal. Dr. Amita Trehan, Professor, Pediatric Hematology Oncology Unit, Advanced Pediatric Center, PGI, Chandigarh. Dr. Atish Narayan Rao Bakane, he's from Indraprasth Apollo Hospital, Delhi. Dr. Harsha Prasad, Assistant Professor, Department of Pediatrics, Kasturba Medical College, Manipal University, Mangaluru. Shruti Toshniwal from United Sigma Hospital, Aurangabad. Dr. Abhilasha Sampagar from KLE Hospital, Belgaum, JNMC. Now it's my pleasure to introduce the guest speaker for the day. He has been our speaker in past, and I'm extremely grateful to him for sparing his valuable time in uplifting the care of children with ALL and others in this part of the world. As you know, he is Director, Clinical Research, Center of Childhood Cancer Research, Associate Professor of Pediatrics, Division of Hematology and Medical Oncology at Children's Hospital, Philadelphia, Perelman School of Medicine, University of Pennsylvania. His areas of expertise include acute lymphoblastic leukemia, ALPS, HLH, and bone marrow transplant. He did his medical school from Eastern Virginia Medical School, Norfolk, Virginia. His pediatric residency was at Children's National Medical Center, Washington, D.C. His fellowship in pediatric hematology and oncology was from Children's Hospital of Philadelphia. He was postdoctoral research fellow in pediatric hematology and oncology at the Children's Hospital of Philadelphia. I invite him to give his talk on childhood acute lymphoblastic leukemia, how to manage in 2022. Thank you. Over to you, sir.
Thank you. Let me share my screen. Can you see my screen and hear me okay?
Yes, sir.
Let's make sure I advance the slides. Okay, so I'd like to thank the organizers for inviting me here today to speak. I'm going to talk today about childhood ALL refining the treatment in 2022, primarily focusing on the US and European perspective, but this applies around the world. As far as disclosures, I get grant funding from a number of companies and sit on the scientific advisory board for a number of companies as well. I think one of the improvements in cure rate in acute lymphoblastic leukemia has been one of the greatest success stories of modern medicine, where 50 years ago, the majority of children who were diagnosed with ALL did not survive. And in 2022, most children can be cured, but we're still not at the finish line yet. And there are a lot of children who still succumb to the disease. Not only has there been dramatic improvement in ALL overall, but there's been certain subtypes of ALL who have had even higher gains of improvement in the last 10 to 20 years. For example, if you go back 20 years ago, the outcomes for children with B-ALL were 10% higher than T-ALL, but currently the outcomes for children with T-cell acute lymphoblastic leukemia are comparable to B-ALL. So what has improved outcome over the past 50 years? First, it was recognized, and a lot of this work was done both in Germany and at St. Jude, that the CNS site is CNS is a sanctuary site, and actually having CNS-directed therapy, despite the long-term morbidity and mortality of that. Augmenting traditional chemotherapy dose schedule and combinations. And then a lot of it's been more recently on development of modern risk stratification, recognizing groups of patients with higher relapse rates that need more therapy, such as older patients, patients with high white blood cell counts, patients who are slow to respond to therapy, and then certain genetic subgroups that have poor prognostic significance. A lot of this I'll get to later. Most of the drugs that we currently use to treat children with ALL, though, although that's changing, which I'll get towards till the end of the talk, are drugs that have been around for a long time and ones that are actually first tested and first developed in the 1950s and 1960s up until currently. Probably the one drug on here that actually has the biggest issue, which is very active, I think, internationally, is asparaginase, because access to this differs based on a lot of countries. But most of these other drugs are pretty much available all over the world. One of the questions that remains is, where can we still do better? While we're improving and curing most kids with ALL, we're not curing them all. I think one of the biggest places that we still have a little bit room to go is in relapse. So the survival for newly diagnosed children with ALL is around 90%, but still children who relapse have less than 50% chance of surviving if you look at all of them. Particularly poor is T-ALL and infants with ALL who relapse, but even B-ALL, of only about half of children who relapse are long-term survivors. Other places that we still need to do better are patients with high-risk subtypes of ALL. These include infants with ALL, where we've really not made any gains in cure rates over the past 20 to 30 years. Hypodiploid ALL is another example, and this is a type that, despite transplant, despite immunotherapies and other therapies, the overall cure rates are still pretty low. I should caution that by saying that we're still learning how much immunotherapies may change the outcome or the long-term survival of children with hypodiploid ALL and infants with ALL. One of the reasons I still think we have a long ways to go is that if you look at newly diagnosed patients with ALL, we've gotten much better at identifying patients that are really high risk, may need transplant in first remission, may have a higher disease biology. But if you look at the numbers, the majority of patients of T-ALL who relapse are patients that we currently think are low risk or more favorable risk. While we know that patients who have N-consolidation MRD that's positive, for example, have poor outcome, that's only 5 to 10% of patients, depending on the backbone that you're using. And in B-ALL, NCI standard risk, i.e. patients who are less than 10 of a low white count, contribute to about half the kids who have treatment failures. Part of this is just a numbers game, where that's the most common group of children. So how do we tackle the problem moving forward in 2022? As I mentioned before, one of the ways we got to where we are now is by intensifying therapy for children with high-risk disease, and reducing therapy for children of lower risk disease and tailoring it to the individual patient. But I think we've gotten as far as we're going to get by rearranging the deck chairs. I think we still have ways to go in improving risk stratification, which I'll talk about a little bit. What I highlighted here in red are the things I'll address today. I think we still have room to go in introducing targeted therapies, and there are a number of trials open across the globe that are looking at targeted therapies. Addressing clonal evolution and chemoresistance is an important topic that I don't think we're tackling quite yet in the clinic, in that blasts at relapse are often very different than they are when patients are diagnosed, and subclones start to arise while you're on therapy. We know this from deep sequencing technology, single cell sequencing, and we're not really using technology yet to identify patients who have a clone that's starting to arise that we could target with novel therapies before they relapse. And I think that'll be a wave in the future, but I'm not going to get to that now. Another thing is incorporating immunotherapies, and this is something that I think is making a huge impact across the globe, and I'll talk about this a little bit. And then the last thing is maybe it's time that we start thinking about the way that we design clinical trials. As I kind of alluded to more, for a long time, the way that we got to where we were was intensifying therapy for a lot of patients. In the children's oncology group, most of our studies over the past, from about 2000 or about 1995 to probably 2010, were based on additional intensification of chemotherapy and a number of consecutive trials through the children's oncology group that were open across North America and part of Europe and Australia and New Zealand. And the long and short of it for all of these trials, for the most part, is they didn't make a dent. Outcomes were relatively the same with all of these intensifications. All you got was more toxicity without extra efficacy. So I think we're moved to a place of not looking at these kind of questions anymore. I think one place that we have made a huge impact that has improved outcomes for children with ALL, but we have a ways to go, is narrowing the racial gap. If you look at US data, the survival for children who are white was significantly better than Black children if you go back 10, 15, 20 years ago. But in 2022, they're comparable now if you look across the US. That doesn't mean that African-American children still aren't disadvantaged in the US or have worse outcomes. It's just that we've gotten it closer. Indeed, Sumit Gupta, who's from Hospital Sick Kids, presented some data at ASH just last month, where he looked at patients who were treated with ALL on COG trials from 2004 to 2019. These data included over 25,000 children and young adults, and also included over 2,000 kids with T-ALL treated on both the ALL 0434, which I'll get to, and 1231 trials. If you look at overall survival in both B and T-ALL, you can see there's significant disparity in outcomes in B-ALL based on race and ethnicity, and which not shown here is also by socioeconomic status in North America. What we were surprised to see is there's actually no difference in outcome in patients of T-cell acute lymphoblastic leukemia. So children who are white or children who are non-Hispanic Black had the identical outcomes, at least when treated with COG therapy in COG institutions on COG trials. We all know that race and ethnicity are social and not biologic constructs. While not biologic, we know that there are poor risk biologies that are enriched in certain subtypes, like PH-like ALL is more common in Hispanic populations. Despite that, even if you adjust for biology, the work that was done here was powered to do that, there was still disparities in B-ALL despite that risk stratification. The one big question we have to ask now is patients with high-risk B-cell ALL who had worse outcome based on race and ethnicity are treated almost identically to patients of T-ALL in the US. They're treated at the same centers. They're treated with almost the same backbone chemotherapy, which raises the question of why children didn't have this outcome disparity. It could be that there's a number of hypotheses for this. It could be there's an enrollment bias in T-ALL. And other cancers such as AML, sicker patients who are diagnosed and are Black are less likely to go on clinical trials. And T-ALL patients are more likely to present critically ill with mediastinal mass and B-cell patients. B-ALL patients are more dependent on maintenance for cure, and T-ALL is more dependent on early intensity. So one could hypothesize that coming to clinic for 3 years to get your therapy, it's easier when you're in the more intensive stage, but when you get in your less intensive stage, for doing it for 3 years can be tough. And maybe that impacts outcomes on B-ALL patients more than T-ALL patients. Another thing that we're looking at right now is Black children have lower ANCs just as part of normative values, and we adjust our chemotherapy based on normal values in white children. And maybe we're holding chemotherapy too much in children with T-ALL, and that's leading them compared to BAL or BAL or Black children compared to white children, and that impacts outcomes in BAL more than T-ALL, where maintenance doesn't matter. One of the ways that we really improved outcomes and understanding of BALL and T-ALL over the past 20 years is by improved biology studies. So my lab is a research lab. We do a lot of preclinical testing and a lot of genomic studies and other studies. And there's a lot of labs around the world that have done this. Through the Children's Oncology Group for the Target Initiative, we've done genetic profiling on well over 3,000 cases. There's a paper that'll probably be coming out in the next few months from the Target Initiative, which is basically summarizing all the genomics that have been done in ALL for the Target Initiative over the past 20 years. This has been a collaboration between St. Jude Children's Research Hospital and the Children's Oncology Group, where most of the sequencing happens at St. Jude. A lot of the patients are there, and then the bulk of patients come from COG studies. What we have learned is you can take patients of both BALL and T-ALL and classify them into different groups based on their primary driving lesions. In BALL, this has been very helpful both for risk stratification and also for identifying patients who could benefit from novel targeted therapies and the development of targeted therapies. And I'll talk about some of those. I think everybody knows that BCR-ABL1 is kind of what changed the paradigm and the way that we thought of precision medicine and targeted therapies. And really, leukemias are what made this first difference, going back to the 90s with imatinib and CML, followed by using imatinib and dasatinib in BALL, where pH-positive ALL, if you look at going back 25 years, was actually had one of the worst outcomes in children with BALL. Now those outcomes are comparable to children without pH-positive ALL. Arguably, other subtypes like pH-like ALL have far worse prognosis in 2020. And by integrating tyrosine kinase inhibitors into backbone chemotherapy, we've not only improved cure rates, but we've also reduced the number of children who need to go to bone marrow transplant, which has significant short and long-term morbidity and mortality. pH-like ALL is a more recently described, I used to say recently described, but it's been a while now, subtype of ALL that has a gene expression profile that looks like BCR-ABL positive ALL, but is BCR-ABL1 negative. And there's been a lot of studies over the past 15, 20 years that have looked at the pH-like ALL subtype to try to understand its biology. It's incredibly complex because it's actually a mixed bag of different patients, 50% of which have CRLF2 rearrangements with or without JAK mutations. 15 to 20% have rearrangements in JAK2 or EPOR, and about 10 to 15% have ABL class alterations, similar to BCR-ABL1, but with different fusion partners. And I think it's complex because if you just look at ABL1, for example, when this slide was made, Sarah Tajan provided it, there were 13 fusion partners that have been identified, and that number is much higher now as we continue to look at patients all over the world of different subtypes of PALL. If you look at outcomes for pH-like ALL in general, they're about 20% lower than the non-pH-like ALL subtype. And this actually happens across age groups where the outcome is different. One thing that's different, though, if you look across age groups, is the frequency or the relative frequency of different types of alterations. And as you get older, they change. In the Children's Oncology Group, we currently use a very complicated algorithm to identify patients with pH-like ALL using basically a card that helps identify the signature. That card can be false positive in subtypes of ALL, so they're ruled out. And then basically uses a number of technologies to drill down on what the driving lesion is. I think we're in a place in 2022 where this algorithm is going to get a lot simpler because we have much better fusion panels. For example, using the Archer technology, but I think also we're at a point now where probably in the next 10 years, we'll be using clinical RNA sequencing and just skipping all these steps. There was some nice data that recently came out of Europe that basically showed that if you do RNA sequencing on patients with ALL at diagnosis, you can replace pretty much all of our cytogenetic fusion testing and other testing just with that singular technology. For some lesions, you probably want to verify them with alternative tech, but at least it makes it so that should be your initial step. Developing CLIA-CAP, or as we call it in the US, laboratory certified RNA-seq is very difficult, but it's getting easier and easier with time. Currently in the COG, based on results of pH-like testing and depending on the lesions, you may be eligible to receive treatment with a tyrosine kinase inhibitor if you have the type of lesion that we think would be beneficial for that, or for a JAK-STAT inhibitor if you have a JAK driving lesion. Because of the wealth of development of understanding of PH-like ALL, there's also been a wealth of new trials that have opened up. Some of them have been going on for a number of years, looking at different inhibitors in patients of PH-like ALL. And the hope would be this will impact cure rates similarly to tyrosine kinase, which did in BCR-ABL1 ALL. I think it's important to highlight, though, that this kind of precision medicine approach, while it is going to probably make an impact in some patients, the total fraction that will benefit from this type of approach is probably relatively small. There's a number of trials that are looking at new targeted approaches for children with higher risk leukemia. I'm not going to go through them all just out of time, but one strategy, for example, is looking at using menin inhibitors in children with relapsed infant ALL. There's a trial that's opening right now in the US looking at Syndax, which is a menin inhibitor in these patients. The biology of looking at menin inhibitors in infant ALL is menin inhibitors basically bind to the MLL1 protein and prevent its fusion with DOT1L, which is important in its biology. And if you actually block this interaction, you would get differentiation of leukemic blasts. In preclinical models with menin inhibitors, the data are pretty striking. Although you have to always give that with a caveat. There have been a lot of drugs that have been tested in preclinical models of MLL rearranged, KMT2-aged ALL that looked very promising in mice that actually didn't translate to success in the clinic. Hopefully this will be an exception to that rule. I think it's also important to note that targeted therapy is not always just picking a bullet or a new drug that's fancy and shiny and new and using it in a new biologic subtype or a described one. This is a lesson that actually came from the Tokyo Children's Group, where their L92-13 trial, which means it ran back in the 1990s. The study question was to reduce therapy down to 1 year. And the way they did that was preserving intensity at the expense of maintenance. So for patients who had more favorable risk disease, who had a shorter intensive therapy phase, they got about 6 months of maintenance. But patients who were really high risk got a total of 9 months of intensive therapy and then only 3 months of maintenance, and everybody stopped at a year. When they initially looked at their data, relapse rates were very high. Event-free survival was very poor, especially in boys. It was 56% when you look overall survival compared to 71%, which was the overall survival rates on prior trials. And one thing of note, though, was even though the overall, the event-free and overall survival dropped, a lot of these kids were salvageable. One thing they did years later is they actually went back and looked at their data. In 2017, they took bank samples from children on this trial and they performed genomics on them to see if there were some patients that actually could benefit from this reduced maintenance. And while numbers were very small, as shown on the slide on the right, boys and girls with ETV6 RUNX1 and TCF or E2A TCF3 PBX1 had 10-year disease-free survival of greater than 90%. So basically, with 1 year of therapy, children of TEL-AML1, ETV6 RUNX1, had a 90% cure rate. On the flip side, children of high hyperdiploid ALL had a 10 years disease-free survival rate of 56%, so half. On most modern chemotherapy protocols using 2.5 to 3 years of therapy, patients of high hyperdiploid and TEL-AML1 have identical outcomes. When you reduce maintenance and total therapy to down 1 year, you actually maintain those outcomes in your ETV6 RUNX1 children, but they dropped significantly in high hyperdiploid kids, which just highlights that there may be certain biologic subtypes that could actually benefit from different levels of treatment, and the ability to get rid of 2 years of therapy in a subset of patients with ALL is actually a huge win. And there are now ongoing trials in Tokyo, which are trying to take this the next step forward and reduce therapy just for certain biologies that may benefit and not need maintenance, similar to TLL. I'm going to shift gears a little bit and talk in a little bit more depth on patients of TLL and targeted inhibitors. And part of that's because right now, which I'll get to at the end when I talk about immunotherapy, there are a lot more immunotherapy options for children with BALL that have actually shown success. In TLL, we're getting closer, and there's actually some exciting data that were presented at ASH and at ASCO this year, and at other meetings looking at immunotherapy in TLL, but those are just starting to get into the clinic. We're learning more about TLL biology. It's far more complex than BALL biology. Patients can't beat nightly bend into different groups where we understand what's driving disease. There's a lot of overlap, and a lot of those genetic lesions don't actually either impact outcome or are immediately targetable. There was some nice data that came out of the Target Initiative quite a few years ago that looked at 264 children of TLL and did genomic profiling, whole exome sequencing and RNA-seq, and found that you could batch patients of TLL into 10 different groups based on biologically targetable pathways. There's considerable overlap in those pathways in that some patients have mutations or alterations both in JAK-STAT, for example, and PI3-AKT-mTOR. But it did show that there's a lot of overlapping biology in TLL that helped us design some targeted therapies that we could try to move forward into the clinic. And I'll go through some of those real quickly. As I kind of mentioned before, we need to understand more about T-ALL biology. One of the problems with some of the genomic work that's been done in TLL by the target group and others is most ALL studies eliminate the most high-risk patients because they make germline DNA from patients who are in remission at the end of induction. So basically use the induction marrow for germline. And if you're not in remission, those patients are excluded. We have an initiative right now that I'm leading in the Children's Oncology Group in collaboration with St. Jude, where we got what's called an NIH XO1 mechanism, and we did genomic profiling on 1,300 children of T-cell acute lymphoblastic leukemia. And we finished the analysis of those patients, and in the next 6 months should have data that we're ready to present, which will be a much more robust genetic map of T-ALL. In this, we were able to include all patients with T-ALL, including the high risk, because we made germline DNA from patients who weren't in remission by a number of different techniques, including sorting blast at diagnosis and removing them to make germline DNA. We're also doing some single cell sequencing on patients with TLL. I won't go into it today, but some of this data was presented by Jason Zhu at ASH this year. And we're starting to understand a lot more about TLL biology at the bulk and single cell level. And I think over the next year is going to be very exciting as we start releasing some of these results. The last 2 clinical trials that were run for the Children's Oncology Group looking at children of T-ALL and T-cell lymphoblastic lymphoma were the 0434 and 1231 clinical trial. The 0434 results have been published, so I'll go through them quickly. This was a study that treated children of T-ALL with a prednisone-based induction. Patients were risk stratified based on things that we don't use as much anymore, but did back then, including early morphologic assessment, steroid pretreatment, early MRD and morphologic response into 3 groups. Patients who were low risk were randomized to receive Capezi or high-dose methotrexate intermaintenance. Intermediate and high-risk patients got that randomization plus nelarabine or no nelarabine, and patients who were induction failures were non-randomly assigned to high-dose methotrexate plus nelarabine. I think it's important to know that nelarabine, which was tested here, is actually a drug that was first described by Gertrude or developed in the 1950s that has happened to be on a shelf for 50 years. The results of that trial, the bottom line, showed that children who were randomized to receive Capezi methotrexate or nelarabine had improved disease-free survival when they were treated for either one of those. Children of Capezi methotrexate also had improved overall survival. Patients treated with nelarabine had a trend towards improved overall survival, but didn't quite cross significance because the Capezi arm so drove the improved outcomes. On this trial, the outcomes, if you look at the control arm, which was the standard in the world at the time of high-dose methotrexate versus no-nel, the outcomes were around 78% for the majority of kids versus 92% versus with Capezi and nelarabine. And most of the improvement was a reduction in relapses. The ALL 1231 trial that was run through COG was looking at the hypothesis that maybe able to move proteasome inhibitors to the front line. The rationale for proteasome inhibition was that they were active in preclinical models. They synergize with chemotherapy. There was data that showed that bortezomib, the proteasome inhibitor we looked at, can overcome chemo resistance, and they may resensitize steroid-resistant TLL cells. Proteasome inhibitors are basically, they target the wastebasket of cells with the ubiquitin protease inhibitor pathway, and they're particularly active on cancer cells that have certain pathways dysregulated, such as NF-kappa B. They were studied in a preclinical trial or early phase clinical trial by Teresa Horton through the Children's Oncology Group ALL07P1 that showed they were active in T-ALL. The results of the 1231 trial were recently released. We presented them in ASH last year, and actually there's a paper right now that's under favorable review and in minor revision at JCO, so hopefully these data will be published in the next month or so. On this trial, we randomized all patients to receive or not receive 4 doses of the proteasome inhibitor bortezomib during induction and delayed intensification. We changed the backbone. We made 2 major changes. One, we changed the steroid throughout it from prednisone to dexamethasone, using a regimen that was shown in the UK 2003 trial, giving it 6 milligrams per meter squared per day for 28 days. There were data from Europe from a BFM trial, AOP BFM 2000, that showed that patients treated with dexamethasone who were randomized compared to prednisone had improved outcomes with TLL dexamethasone if they were steroid good responders. But we don't use a steroid prephase in the COG, so we changed the backbone for everybody. Patients are risk stratified primarily based on MRD. One of the reasons we changed the backbone, both adding steroids and the other major modification we added was giving a couple extra doses of asparaginase, one in induction, one delayed intensification, was to remove cranial radiation. On the 0434 trial, while outcomes were excellent, 90% of children received prophylactic radiation. On 1231, we only radiated children who had CNS3 disease or patients who were very high risk, and this was based on an induction consolidation response. We initially hypothesized that'd be about 10% of patients, but with intensification of therapy, that was actually less than 5% of patients. For patients who were very high risk, we also tried using 3 intensification blocks. And at the end of those blocks, they were developed by the BFM. If you're MRD positive, you came off study and went to transplant. And if you're MRD negative, you can stay on therapy on chemotherapy. Lymphoblastic lymphoma patients basically were treated the exact same way. The only difference is they were risk stratified differently, but they participated in the same randomization. We looked at the primary endpoint, and these data were recently updated. So we presented it to ASH to 4-year data. So these are now mature data. If you just looked at arm A versus arm B overall, there was no improvement in event-free survival with the addition of bortezomib. If you looked at overall survival, there was a trend towards improvement on the bortezomib arm, but it did not cross statistical significance. And these data are mature enough now that they're not going to. I think one thing we're highly unexpected to see and very surprised was outcomes for children of T-cell lymphoblastic lymphoma saw a significant benefit in both, an improvement in both event-free and overall survival. Indeed, if you look at 4-year EFS in T-cell lymphoblastic lymphoma patients, it was a 10% improvement in event-free survival. Sorry, they're auto-advancing. And if you looked at overall survival, it was a similar 10% improvement. In contrast, when you looked at children of T-cell acute lymphoblastic leukemia, there was no benefit whatsoever from the use of our proteasome inhibitor. This was quite surprising. There's been a lot of work over the past 20 years to kind of harmonize therapy for children of TALL and T-cell lymphoblastic lymphoma with the thought that they're actually a spectrum of the same disease. But there was a clear difference in outcome with a proteasome inhibitor in TALL versus T-cell acute lymphoblastic leukemia. The other thing, which is a little bit hard to see here, is that if you look at the outcomes on the control arm in T-cell lymphoblastic lymphoma on 1231 in TLL versus T-cell lymphoblastic lymphoma, the TL outcomes are actually pretty comparable to prior trial. And typically, T-cell lymphoblastic lymphoma does better on TLL on our phase 3 trials, at least in the Children's Oncology Group. But here they did actually markedly worse. On the control arm, their EFS was 76% versus 81% for the TLL patients, and far worse than on 0434. We intensified therapy and changed from pred to dex to get rid of radiation in TLL. Radiation actually isn't part of the normal backbone in patients with T-cell lymphoblastic lymphoma. And maybe that was a mistake in the T-cell lymphoblastic lymphoma patients because they didn't benefit from that. So by changing the therapy to dexamethasone, TLL patients seem to benefit from that. T-cell lymphoblastic lymphoma patients don't. And then TLL patients don't benefit from proteasome inhibitors, and TLL patients did, showing 2 major treatment differences between these groups that we are now doing some preclinical work to try to tease out why. And this may impact how we treat these patients, and we may have to tailor our therapy differently and treat them differently in future trials. As I mentioned, one of the other big questions we had on this trial, one of the other major goals, was to eliminate prophylactic cranial radiation. This very busy slide is looking at outcomes based on all the different treatment arms on 0434 and 1231. And this is a subdivision comparison where we're only looking at patients who received radiation on 0434 and did not receive it on 1231. This is not all patients. But the bottom line here is that we were able to successfully eliminate radiation in these patients without a worsening outcome, either event-free or overall survival, or an increase in relapse rates, overall relapse rates. Another question that we looked at on the 1231 and 043 trials were CNS status. In patients of B-ALL, there's been a lot of data coming out showing that patients who have CNS2 disease, or they have detectable blasts on CSF cytospin, but a low white count in their CSF, less than 5 cells per microliter, are shown to have worse outcome in B-ALL. On T-ALL, this is some nice work presented by Nathan Gasai that was presented at SIOP and ASCO and is getting ready to prepare for publication. There's no difference in outcome for patients of CNS1 or CNS2 disease. And this was on 0434 and 1231, regardless if you received or did not receive radiation. I'm showing the 3-year data here for 1231. They need to be updated, but it shows the same basic thing. This is also the same regardless of arm. So the high-dose methotrexate arm, the easy methotrexate arm, nelarabine, without nelarabine, with bortezomib, without bortezomib, however you slice it, patients with T-ALL who have CNS2 disease have identical outcomes to patients with CNS1 disease and should be treated the same. In contrast, and if you drill down on even more, because the question we sometimes get asked, some CNS2 patients are not able to be low risk. If you just looked at our intermediate risk patients on 1231, the CNS1 and CNS2 patients received identical therapy and had similar outcomes. In contrast, CNS3 patients continue to do worse despite intensive therapy, despite novel therapies, despite cranial radiation, and we need to do something different for those patients. Based on these results of those 2 trials that I just mentioned, in North America and in most of Europe, the standard has transitioned for T-ALL to pretty much use a dexamethasone-based induction, followed by 0434-like therapy, using an augmented BFM consolidation. We use a single intermittent phase of Capezi. We risk stratify based on MRD. And in the US, we give nelarabine to all of our patients. I know that's not available at some parts of the world, and that actually is one thing that differs between our approach and the Europeans and all over the world. Nelarabine, even though it's a 60, 70-year-old drug, is quite expensive. And there are some groups, I know there was actually a generic nelarabine that was just developed and came out for clinical use from India, and one that's being developed in Ireland as well. And hopefully that'll make it much more affordable. And then we'll be able to give it to more children of T-ALL because it does seem to improve outcome, at least in T-cell leukemia patients. As far as where we go next, there are a number of targeted therapies that are looking very promising in patients of T-ALL. One of the first targeted therapies that was really studied was looking at JAK-STAT inhibition for T-ALL. And this was really driven based on some data looking at ETP versus non-ETP ALL. Some of the initial work came out of my lab, where we looked at JAK-STAT signaling in patients of ETP and found it was typically dysregulated in ETP ALL, regardless if you had an activating mutation in a JAK protein or gene or not. When we treated murine PDX models with JAK-STAT inhibitors, ETP ALLs were exquisitely sensitive to this, versus non-ETP ALLs. Most of them were not, although some of them did have dysregulated JAK-STAT signaling, which I'll talk about in the next slide. Ruxolitinib, which is a JAK-STAT inhibitor, has been studied in a lot of different contexts. And currently in St. Jude, they're incorporating ruxolitinib into therapy for de novo ETP and MRD positive ALL, and we anxiously await the results of that trial. As I kind of mentioned earlier, the BFM actually uses a steroid prephase to look at steroid sensitivity in patients of B and T-ALL. They use it for risk stratification. And in T-ALL, if you're steroid responsive, you get dexamethasone. And if you're steroid resistant, after that prephase, you go on a prednisone-based phase. And they found that about 1/3 of patients have intrinsic steroid resistance. Lauren Meyer, a very talented postdoc in Michelle Hermiston's lab, has been looking at steroid resistance in T-ALL, particularly wanting to understand, we know that ETP ALL is intrinsically corticosteroid resistant and also has dysregulated JAK-STAT signaling. Is there a group of non-ETP ALL, T-ALL patients that are also corticosteroid resistant intrinsically by same biologic pathways? And for this, basically, we performed RNA-seq and nanostring technology on a large cohort of T-ALL samples. And she identified a nanostring signature that actually was distinct for ETP, but also showed the same signature was found in patients of non-ETP ALL and enriched in the ones that were MRD positive. Using the signature and applying it to a larger cohort of patients treated on 0434, she could show this signature actually predicted who would be MRD positive at the end of induction. This was then validated on 1231 patients, and we're currently taking that EXO-1 cohort, which did RNA-seq on 1,250 patients I mentioned before, to see if the signature actually correlates with outcome. One may ask, why is it important, or why is it helpful to know if a patient's steroid resistant when they walk in the door, if they'll later be, if you can just identify this by MRD positivity? And I think the 1231 trial and the data from Europe kind of highlight that if we know somebody's intrinsically steroid resistant, maybe we shouldn't be treating them with an intensive steroid backbone from the get-go with dexamethasone. Maybe those are patients we should be treated with prednisone. Because one of the reasons that some of the outcomes were worse on 1231 than the predecessor trial was there were far more toxic deaths when treating with dexamethasone versus prednisone. So while it may reduce relapse-free survival or improve it in some patients, it may not be a benefit in all patients, something highlighted by the T-cell lymphoblastic lymphoma patients on that trial as well. Another drug which I think is actually very interesting for T-ALL because it's so easy to integrate in our therapy is dasatinib. So Jean-Pierre Boquin's lab looked at drug sensitivity profiling on 68 cases of ALL of 60 drugs. This is data he published in Blood in 2017. And he initially looked at 24 TLLs and had a validation cohort. And he found that 30% of TLLs were sensitive to dasatinib. And when he looked, they did not have ABL fusions. And he also looked and they didn't respond to other TKIs. So this was a dasatinib focused type effect. And he hypothesized this may be by dasatinib also targeting SRC. And he found that SRC was elevated in dasatinib sensitive samples. And then he used a SRC specific inhibitor ex vivo and in PDX models was very effective. This data was then taken to the next step, recently published by Jun Yang's lab, that also profiled using preclinical modeling a large cohort of patients with T-ALL with a number of different drugs. One of them on the panel was dasatinib. And he found that 44% of children and 70% of adults in this group were sensitive to dasatinib. Using RNA-seq and an algorithm that they developed there called NetBid, which integrates genomics with pharmacotyping, they found that there was upregulation of the pre-TCR LCK pathway. LCK is a member of the SRC family of kinases. So it looks like there's a subset of TLL, maybe almost half of pediatric TLL, that has upregulation of LCK that leads to exquisite sensitivity to dasatinib. If we could find a nice biomarker from this at diagnosis to actually be in a clinical test, integrating dasatinib into our frontline therapy would be very easy, or using it as a relapse drug in patients who benefit would be very important. So I think we now have an old drug that we know is effective in probably 50% possibly of patients with TLL. Of course, this needs to be validated in prospective trials. We just need to identify who those patients are in real time, because it's not a simple thing that can be done just by looking like ABL fusions like you can in pH positive or pH like ALL. Another area of interest is targeting the apoptotic machinery. Some older data from two nice publications looked at targeting BCL2 in patients with T-ALL. These data basically showed that patients with ETP ALL are more addicted to BCL2 versus patients with more mature TLL are more dependent on BCL-XL. BCL2 you can target just with venetoclax, a drug that's used very commonly in lots of different types of leukemia now. This was shown in preclinical models. And patients with non-ETP TLL aren't BCL dependent, so they're not going to probably benefit from venetoclax. But if you give venetoclax with low-dose navitoclax, you can actually target BCL-XL as well. And that was actually very effective in preclinical models. There are a number of trials going on looking at this. Wendy Stock recently published some results of an early phase trial, just came out this year in Cancer Discovery, where on this trial, they gave patients venetoclax for 2 days, followed by venetoclax and low-dose navitoclax, and they allowed them to get chemotherapy. It only included a small number of children, but the overall response rate was very high in PIDS, 86%. If you look at the overall results of the trial and look at T-ALL and adults with TLL with relapse disease, the results were very promising with the Vin-Nav combo in these patients. The Europeans have a trial right now. They're kind of a basket targeted trial for the ITCC group, the HemeSmart trial, and this has multiple arms that include VIN and some with VIN-NAB. There are also studies in development in St. Jude's, and currently in the Children's Oncology group, we're developing a trial for relapsed T-ALL to add venetoclax and navitoclax into the backbone to get more data in a larger cohort of children, with the hope that this looks effective, we may be able to move VIN-NAB into the front line. One thing that I won't mention or show here today, but I think was mentioned in that paper of June Yang's, there are, if you look at the patients who are dasatinib sensitive in that paper, a lot of them are resistant to venetoclax and vice versa. So there may be a preclinical signal that there's a subset of patients with T-ALL that are sensitive to dasatinib and a different subset that are sensitive to venetoclax, and they may not be overlapping. And it may be that the future of T-ALL will be doing biomarker studies at diagnosis. Half your kids are going to get a dasatinib-based added on backbone, and half your kids are going to get VEN-NAV added to your backbone. And that way we'll be able to get a novel therapy to the majority of kids who are higher risk disease. But I think time will tell if that approach will be feasible and effective. Another pathway that is promising in T-ALL is targeting cyclin-dependent kinases. There's a number of different cyclin-dependent kinase inhibitors, including CDK4/6 inhibitors. This is some work that came out of collaboration between my own and Yannis Iafanis' lab, working with Michelle Rates and Bill Carroll, where cyclin-dependent kinases are frequently dysregulated in T-ALL, and CDK4/6 inhibitors, including ribociclib and palbociclib, are one way to target those. These data were just published in Hematologica last month, where we took mouse models of B and T-ALL and treated them with different CDK4/6 inhibitors alone and in combination. We found in T-ALL in particular, the CDK4/6 inhibitors were quite effective. There were some in vitro data that showed that conventional cytotoxics can actually antagonize CDK4/6 inhibitors, which raised a lot of concern moving these drugs into the clinic if they're given at the same time. So we decided to model that in our mouse models and actually found that was not the case in mouse models. So even though in vitro, CDK4/6 inhibitors, when given at the same time as conventional cytotoxics, may inhibit the interaction of cell cycle-specific agents such as vincristine, this was actually not the case in vivo, which made us very comfortable to move forward with a clinical trial that Elizabeth Rates chaired that was presented at ASH this past year and should be published soon, which looked at adding in a CDK4/6 inhibitor, palbociclib, onto a VBLD-based backbone for patients with relapsed and refractory B and T-ALL. Not allowed to talk about the efficacy data today. Hopefully that'll be released soon, but from a safety standpoint, it was very safe and effective. It was very safe. As I kind of mentioned in the beginning, in T-ALL right now, we're still in the world of targeted therapies, although we're moving into the immunotherapy era quickly. In B-ALL, starting about 10, 15 years ago, really immunotherapies became the next great wave of improving outcomes in B-ALL. And there's lots of different immunotherapies that are out there. One of the reasons that immunotherapies are very promising for B-ALL is you can wipe out your normal B cells and not cause significant morbidity and mortality versus other cellular populations. And there are antigens on the surface of B-cell lymphoblasts that are only on B cells and not other populations in your body as well, like CD19 and CD22. Everybody knows there's a number of B-cell directed immunotherapies that are out there. There's blinatumomab, a bispecific T-cell engager. There's inotuzumab, a CD22 monoclonal antibody that's conjugated to bleomycin. There's rituximab, CD20, naked monoclonal antibody. There's a dozen other drugs that are out in the clinic now. There's a lot of them that are in clinical development in adults. And then there's chimeric antigen receptor T-cell therapy. I think important to highlight that one of those earlier slides that I showed that shows most of the drugs that we use in ALL are ones that developed from the 50s to the 70s. Yet in the past 5 years, there's been 3 immunotherapies that have been FDA approved and EU approved for children with relapsed and refractory B-ALL and adults. These include inotuzumab, blinatumomab, and tisagen-lechlecel, the first CAR T-cell therapy or gene therapy approved for regulatory use. Most of the data in inotuzumab were initially published in adults. This is some data initially published in the New England Journal of Medicine, randomizing patients who are adults from inotuzumab versus standard therapy, showing an improvement in progression-free survival. We have much better outcome data in kids, which I'm not showing here today because they just came out. Maureen O'Brien led an early phase trial in pediatrics for the Children's Oncology Group that were just published in JCO this month that showed an impressive efficacy signal in relapsed B-ALL. Blinatumomab was also first studied in adults, followed by studies in early phase clinical trials in kids. Some of the data are shown here, showing it was a very active drug. And based on this, we in the Children's Oncology Group, in a trial that was run by Pat Brown, that was published in JAMA just this year, randomized high-risk patients, a certain group of patients, I won't go for the risk stratification because it's complicated, to receive chemotherapy, backbone therapy, or on RMB to also get blinatumomab. And as you can see here, blinatumomab in this group of patients, which is depicted by the blue line, had markedly improved disease-free and overall survival. So very impressive results, changing the standard of care for first relapse BLL, at least in a subset of high-risk patients. Chimeric antigen receptor T cells are something that have made huge waves in the field of cancer therapy. I'm not going to go through all the cancer CAR T cell biology based on time. CAR T cells basically are genetically reprogramming a T cell so it binds to and can attack a tumor antigen. The concept of doing this goes back to the 80s and 90s, and initial trials failed. It wasn't until co-stimulatory domains were added into CAR T cells such as CD28 or 4-1BB that allowed the T cell to bind, and then after binding and killing cell, it would lead to the T cell to grow and proliferate instead of undergoing apoptosis. And this is really what the big breakthrough that led to improvement in outcomes of CAR T. The first preclinical, one of the first CAR T cells to get treated in the clinic was tisagenlecleucel, which was initially developed at UPenn and CHOP. This was some of our early data looking in mouse models, PDX models, where we randomized mice to receive tisagenlecleucel or vehicle control. These are work that were done by John Fish and Dave Barrett, showing that the mice would go into remission very quickly with tisagenlecleucel, at least of some ALL models. This led to rapid early phase clinical trials, which eventually led to its FDA approval. At Penn and CHOP, we've treated now well over 500 patients with different malignancies, with CD19 directed CARs, plus other CARs. And we continue to see a very high complete remission rate, relapse-free survival, and overall survival with treatment in patients with highly refractory disease. It gets into CNS, it cures the CNS. There's been a number of studies that have been published on this. The Eliana trial was the trial that led to FDA and EU approval for tisagenlecleucel, which showed significant relapse-free survival in high-risk patients of BALL. There's also, not trying to just show the CHOP and Penn side of the story, there's been a lot of CD19 CARs that have been developed by a number of groups all over the world. One of the first ones to be developed was Michelle Sadelain's group from Sloan Kettering, who has outstanding outcomes with their CD19 CAR that uses a 28 zeta co-stimulatory domain versus the 4-1BB that we use at Penn. I think there's pros and cons to both of those for different CARs and different diseases. And I think there's been a lot of different CD19 CARs, CD22 CARs that have shown significant outcomes that I don't have time to get into today. Based on the promising data in relapse trials in patients with BALL and the Children's Oncology Group, we currently have decided to move immunotherapy and went all in to the front line. This is a slide from Mignon Lowe and Elizabeth Rates. We currently have 3 trials from the COG. One is taking standard risk BLL patients, a subset of them, and randomizing them to receive or not receive blinatumomab on a higher risk backbone, or for standard risk average, just randomizing them to receive blinatumomab. This is a study being run by Sumit Gupta and Rachel Rao. Jen McNear and Maureen O'Brien are running a trial in NC high-risk patients where a subset of them are getting randomized to receive or not receive inotuzumab. And then Shannon Maud is running a trial where we're looking at adding tisagenlecleucel non-randomly to very high-risk patients with BALL, primarily based on inconsolidation MRD positivity. So the majority of patients in COG studies now in the front line are getting have access to possibly B-cell immunotherapy. There is a subset of patients who are basically saying the battle is won. Their outcomes are too good. We're no longer powered to ask a question. And then there are a small group of patients that are eligible for targeted therapies in BALL. Back to the thing I'll close out with now and finish up my talk is talking about a little bit about immunotherapy for T-ALL. As I mentioned, immunotherapies have been highly effective in patients of BALL. In T-ALL, we actually haven't made the same degree of inroads over the past 15 years for a number of reasons. One is target. We need to find targets that are on T cells, but not on other cells. And that really impacts toxicity as well, where a patient can't live without their normal T cells. There's an issue of fratricide where the CAR T cells or other immunotherapies will attack the cell they're based on. This is not an issue for monoclonals, but it's an issue for CARs and BiTEs. And then the other issue, which gets to be tricky, is eliminating blasts from products. There was some data that came out with some B cell CARs from Penn showing that there's a low level risk that you actually could transduce or transfect your CAR T cell and a leukemia cell with your CAR, which could make you resistant to the CAR. And you don't want that to happen. And that's a bigger issue in T-ALL, that you need the sufficient systems to sort out blasts from your product. Patients with T-ALL often, the ones that are eligible for CAR T cell trials, often have refractory disease. They often have circulating T cell blasts, and you need to remove them, and which can be a very challenging thing to do. My group and others looked at different cell surface markers to see what were good targets in T-ALL. I think the biggies that are right now out there are probably CD5, CD7, and CD38 for a number of reasons. They're pretty much ubiquitously expressed across T-ALL samples, both at diagnosis and at relapse. Our lab has been really focused on looking at CD38. One of the reasons we like that is it's highly expressed on most TLLs. It's consistently highly expressed as patients are treated with chemotherapy and relapse. In contrast, patients with B-ALL, if you look at CD38 expression, it actually goes down just to treatment of chemotherapy. Mechanistic data I won't show here today suggests that TLL is really addicted to CD38 in contrast to B-ALL. Based on this, we tested the naked monoclonal antibody daratumumab in preclinical models of T-ALL and found it was very highly effective. This surprised us a lot, actually, since it's a naked monoclonal. Most naked monoclonals do not work in preclinical PDX models because they have no immune system, so you have no ADCC. But we found that this was due to direct cell lysis by daratumumab. This led us to work with Janssen in an early phase trial. It had a safety and an efficacy stage. It initially included B-ALL patients. Not surprisingly, they didn't respond. It also included a T-cell cohort, and if it was found to be safe and effective at an early signal, it moved to a second stage, pretty standard stage 2 design, which it moved forward in. I can't talk about the efficacy data today. The study is closed. The data are being submitted to ASCO for presentation, and so they should be available this summer. I can say the data are encouraging enough that we are in the process of developing a frontline phase 3 trial right now through the COG and some European partners with the Altogether group and possibly even the IBFM as well. It's being discussed at the end of this month, of randomizing patients to receive or not receive daratumumab as part of frontline therapy for high-risk patients based on the results of the preclinical trial that I can't talk about quite yet. This is the schema for the trial that's in development, where basically patients who are MRD positive at the end of induction will be randomized to receive or not receive daratumumab. Patients who are persistently positive at the end of consolidation will be considered treatment failures and can non-randomly get daratumumab. There's been some very exciting data that are coming out from Europe that have looked at preclinical models looking at targeting CD47 in patients with T-ALL. There are a number of CD47 monoclonal antibodies. This is the CD47 is a don't eat me signal on macrophages. It looks very promising. And what I think is very nice is there's some data combining CD38 and CD47 MOABs in mouse models showing synergy with this combination. There's currently an anti-CD47 relapse trial that's in development for the COG, and one could possibly envision, if we move daratumumab to the frontline in T-ALL and it's effective, that the next trial could be adding in a CD47 monoclonal antibody in patients with T-ALL getting randomized to receive on a CD38 positive backbone, a CD47 inhibitor alone, as baby steps in T-ALL to try to reduce some of our intensive cytotoxic chemotherapy and enter the immunotherapy age. There's also a number of CAR T cells that are in development for children with T-ALL. This is just looking at some preclinical data. There's very exciting data looking at CD2, CD5, CD7 CARs. I won't show it today. Our lab has been looking at CD38 CARs in preclinical models, and that's a CD38 CAR trial that will be opening up this year for patients with relapse and refractory T and ALL and AML. Very exciting is now we're starting to see some data get published about using CAR T cells in patients. These are some data from China, which looked at a donor-derived CD7 CAR T cells. So by donor-derived, basically, whoever's going to be the donor for transplant, they also collected T cells from that patient and made a CAR of that. So they looked at 20 patients. 12 had a prior stem cell transplant. They went back to the donor. 8 were stem cell transplant naive, and they collected CAR T cells from the future donor. And then they used those CAR T cells followed by transplant. Importantly, 19 of the 20 patients responded. They went into remission with this CAR T cell. While the follow-up is short, this was just published in JCO this past year, the drug was well tolerated. And it did have an impressive CR rate, and some of them are at least durable out to 6 months. It was also well tolerated. GVHD, there was some grade 1 to 2. They treated it with a unique combination of dexamethasone and ruxolitinib. Even in patients of haplodonors, graft-versus-host disease was controllable. They saw some low-grade CRS and neurotoxicity, but no high-grade. Probably the bigger toxicity they saw, which was not too surprising, was some infections. They did have some severe infections, including one fungal pneumonia death, although that patient may have gone into the CAR T cells with fungus. They did have one CD7 negative relapse. And I think the most fascinating thing about this study was they actually found that in patients treated with this CAR, they had recovery of CD7 negative normal T cell repertoire. So there's actually very small populations of CD7 negative normal T cells that are circulating that most people don't even notice or even study. But when you treat with a CAR that's targeting CD7, that population outgrows. Now, it's small numbers and you have to see how functional they are, but they actually did some ex vivo functional study and found that these CD7 negative T cells were effective against CMV and fungus ex vivo. So potentially, while people thought that CAR T cells would only be in T-ALL used as a bridge to transplant, there may be patients who actually don't need to use it as a bridge to transplant and could be cured. And the last data I'll show today is my lab has been in collaboration with Beam, which is a therapeutic company in the United States. This is work done by Caroline Deora in the lab in collaboration with Sarah Tajan, who's doing similar work in AML, where we've been developing an off-the-shelf CAR T cell for T-ALL using cytosine base editing. Basically, the concept of using an off-the-shelf CAR is kind of promising, even though we're developing a CD38 CAR, which will be one that's auto, because you don't have to worry about separating the blast from the product. Patients with T-ALL get sick. It'd be nice to have an off-the-shelf product you could use right away. The off-the-shelf CARs do require a lot of genetic modifications, though. You need to prevent fratricide, rejection, GVHD, and we're now learning that many gene-edited products using traditional editing techniques like CRISPR-Cas9, TALEN, zinc finger nucleases through double-stranded breaks can actually cause off-target damage and off-target effects. Recently, there was an allogene clinical trial in sickle cell disease that was paused in the United States because of this concern. So more modern base editing techniques may overcome this. And cysteine base editing is one of these. Basically, you use acididine desaminase, vinicase Cas9, and uracil glycosylase, which allows you to convert CG base pairs to TA base pairs at a target site. So we deleted CD7 to avoid fratricide, TRAC, or the TCR alpha chain, to prevent GVHD, CD52, so we could use camppath and conditioning if we wanted to, and then PD1 we knocked out to help CAR T cells invade immune inhibition. We compared cytosine base editing to Cas9 with increasing number of edits. And we basically found that if you use Cas9 and edited out a lot of different genes, it decreased T cell proliferation and activity. So it went down to 40% when you got to 3 edits. And there was no difference in base editing. Using next-gen sequencing, Euditis assay, we found that with Cas9 treated cells, 0.4 to 1.6 of them had on-target or off-target translocations, and none were seen in CBE. When we actually looked at RNA, we also found in a subset of patients or a subset of cells treated with Cas9, they ended up developing chromothripsis, which was not seen in cytosine base editing. We looked at RNA-seq. Cas9, you had upregulation of over 69 different genes that were off-target, including genes that are involved in pathways such as apoptosis, programmed cell death, TP53, and cytosine-based editing. We saw no off-target genetic effects of this as well. And then importantly, we took these cytosine-based edited CD7 CAR T cells and treated them in preclinical mouse models and found that they were highly effective in our T-ALL mouse models. And based on this, we're in the process of developing a clinical trial that we also hope will open in the next year using these off-the-shelf CAR Ts. So hopefully in the next year at our center, we'll have a CD38 CAR, auto, and off the 7 CD7 CAR. But we'll have to see during the pandemic if they actually are able to open the next year. Just to highlight that I just talked about a very US-centric version of some of the CAR T-cell stuff, with the exception of the Chinese trial. There's a lot of different trials for CAR Ts and immunotherapies that are opening in T-ALL in the US and North America, China, India, across the globe. And I think it's going to be a very exciting thing to look at over the next couple of years. And then lastly, the question is, where do we kind of go from here, and some just questions to toss out there. Currently, we decide who goes on which trials based on outcome, but should we be using biology to impact that more? So right now, as I mentioned, patients are binned into groups based on the affected event-free survival in the control arm. But in precision medicine, it really raises the question is, maybe we should just take patients regardless of baseline EFS and put them on drugs they may benefit from. And then in immunotherapies and transplant, the same things as well. Some biologic subtypes are probably more likely to benefit from immune therapies and or transplant than others. We know hypodiploid ALL doesn't benefit from stem cell transplant, and maybe we should allocate their patients to different therapies based on the expectation they'll respond to it, based not just their survival chance. I think one thing, we've gotten more cautious with time, and can take more baby leaps, and maybe we should take bigger leaps in high-risk groups, children like infants. We get pushback all the time in the US that if we want to try a new combination, we have to try each drug one at a time. We can't just try 2 or 3 new drugs at the same time, because you can't assess the impact of the individual agent. But if we had the same approach in the 50s and 60s, Hans-Jörg Riem and Don Pinkel spent 8 years isolating the individual impact of single agents on each trial, we'd never be where we are today. The question in the CAR T-cell world is, there's a lot of work that sequentially targets CARs, uses CARs. We have to target 19 first, then we use CD22 CARs, then we can do a combination of CD19-22. Then we test CD20 CARs as monotherapy, then we can trust a triple CAR, with the thought is you don't want to target agents that may not work. And then you have to start all over again when you have a new manufacturing process. But maybe we should try to make some more leaps. We need to do better in infants and hypodiploid ALL. As I kind of mentioned at the very beginning, the biology of newly diagnosed and relapsed ALL are different. And it raises the question, do we lose good agents of traditional relapse approach? Are there drugs that don't work in relapse that actually be highly effective in newly diagnosed patients? And are there drugs that should be tested in relapse and stop there because they're never going to be effective in the front line? For example, MAP kinase inhibitors. You only have MAP kinase alterations that are enriched in leukemia relapse for the most part. How do we prioritize all the novel CARs being developed across the world when there's every place under the sun has a CD19 CAR they're testing right now? How do we decide which one's the best to move forward? We need more basket trials in ALL, these concepts of opening a trial with multiple drugs. And the other thing we learned in the 1231 trial is, how do we plan the next trial while we're guessing the current trial results? That trial got hurt a little bit when the nelarabine results came out showing it was effective, and we didn't have nelarabine on the 1231 frontline trial. Outcomes are getting better. We definitely need more international collaboration, both to increase patient numbers and diversity, because we know that all patients of all different races and ethnicities are not going to respond to the same drugs the same way. And with that, I probably ran over, but I will conclude. Thanks again for inviting me to talk. Went way over about it.
Thank you, Dr. Tichy, for that wonderful update. We have our Indian faculty here. They can open their videos, and we'll start with the questions. Dr. Amita Trehan.
Good. Yeah. So, hi, that was very wonderful talk, and we really enjoyed it. So I have a very simple, basic question, that when you're talking about steroid resistance, right? So even what we are doing in India these days, we're giving pro-phase steroid, and then depending on the response, we are going on to the risk stratifying, and of course, depending on the other things. So we do know that these differences exist between DEXA and prednisolone, and the toxicities, etc. And you also mentioned that there's a high percentage of patients which are intrinsically steroid resistant, especially T cells. So if a patient turns out to have a poor prednisolone response on like day 8, would then shifting to DEXA be an option? Or because it's possible that they may respond to DEXA, it's a different steroid with more intensive. Would that be an option?
That's a great question. I think that's one of the questions that was kind of left from some of the European trials as well. If you look at the BFM study where they randomized patients to Dexapred, they didn't find that the prednisone poor responders benefited from Dex. And I think that our data on, which some of the stuff I didn't show today on 1231 are showing the same thing. So if you look at the early T-cell precursor ALL treated on 0434 versus 1231, where we used a pred versus a dexamethasone-based backbone. On 0434, the ETP patients actually had the same outcome as non-ETP patients. That's been the dogma we've been talking for a while. ETP is not worse prognosis, treated the same. On 1231, which we haven't shown yet, so very confidential data, the ETPs did worse. If you look at it, it's because they did not have the clinical benefit of the dexamethasone because they're steroid resistant, but they got all the toxicity. And by having that change, you basically just added more toxicity without decreasing relapse, which I think is just another example that patients who are steroid resistant to pred are probably steroid resistant enough to Dex that it doesn't offset the toxicity. So there may be, if you look at relapse only, the patients who are pred sensitive or resistant, they probably both have decreased relapses on Dex. The difference is the margin of that is less than what is needed to outweigh the toxicity of the dexamethasone. So it makes it not worth it. I still think they need steroids. So even the ones that are prednisone relatively resistant probably clearly benefit from steroids. It's just not enough to justify intensifying those steroids.
Thank you. Dr. Akanksha.
Thank you, Dr. Tichy. That was really comprehensive. I really enjoyed it. So my question to you would be is, how would you approach a patient of ALL who has an induction failure? And would that approach be different if it's a T-ALL versus a B-ALL?
Yes. Two things. One is big picture. So T-ALL, so what I didn't show today also was our outcomes on our very high-risk group in T-ALL on our last trial. And a couple highlights from that. One, induction failure T-ALL, if you become in consolidation MRD negative versus if you stay MRD, it doesn't matter. So basically, day 29 response is totally not prognostic in T-ALL. I shouldn't say completely not prognostic. Patients who are MRD negative at day 29 have excellent 95% survival. Patients who are MRD positive, regardless of the degree of MRD positive activity, at least on the dex-based pathosome, do well if they're in consolidation MRD negative, which I think you have to have that later time point. But my bias now is if you're an induction failure of T-ALL, continue on standard therapy and reassess at end consolidation to see if you're in an MRD negative remission or not. The other thing we found on 1231 was that I showed on that slide that for patients who still had positive MRD, we hit them with very intensive therapy, and if they went MRD undetectable, they stayed on chemo. They all died. So taking patients who were MRD consolidation positive and intensifying chemo to make them MRD negative and just treating them with chemo, they all relapsed. This number was small, but it did not work. In contrast, the patients who were in consolidation MRD positive who got intensive chemo and didn't respond to that and then went to transplant, they were all cured. It's very striking. It was 90% cure in the transplant group and 10% cure on the MRD undetectable and non-transplant. Tells two things. One, intensifying therapy isn't the solution. And two, patients who have persistent disease at the end of consolidation after an intensive backbone. So if you're giving them a dex-based backbone, if you're giving them all the drugs that we give nowadays, those patients need a transplant to be cured. You're not going to cure them with chemotherapy. That could get all rewritten now, of course, in the immunotherapy age. We may find that if you use immunotherapy in those patients, it may actually prevent you from needing a transplant, but I think the standard is moved that you need to do that. B-ALL patients, in contrast, who are slow responders, induction failures, they're different. Early response in B-ALL is highly more prognostic, and those patients need alternative therapies, more aggressive treatments, likely a transplant. I think for the B-ALL patients, it's a little nuanced because the question is, were in the induction failure because they were a PH-like ALL that you didn't know about, and now you have a targeted agent that you can add into the therapy, Are they a hypodiploid patient who has an induction failure, where them you might just want to take on immunotherapy because we know that transplant isn't going to fix them anyway? So I think it really depends on, it's not probably a one size fit all type thing, but we do know those patients do poorly with traditional approaches and need something different.
Thank you. Dr. Shripad?
Yeah, thank you, Dr. Tichy. So, when I was doing my fellowship at St. Jude's, I thought that we have conquered ALL and it will get away with, nearly 100. Dr. Poo used to always say 100% cure rate with simple therapies. But looking at this lecture now, I feel that it's becoming more and more complicated now to take care of ALL. And in the short run, I think we'll have hundreds of different subclasses of treatment for ALL. So my question, for example, in India, which we feel here, rather than relapse, that we have a lot of patients which we lose because of toxicity, So considering that, and with the fact that if you see the data of using antibody therapy in relapse setting, post-induction, rather than giving intensive chemotherapy, you think it will become using antibody therapies will be more norm, especially in our type of, where we have more infectious related deaths, will be more easy to use non-intensive therapy using the antibodies therapies rather than intensifying the post-induction therapies?
It's a great question. I think overarchingly, the answer is probably going to be yes as we continue to move forward. I think that if you look at the 1331, even though that was a relapse data that Pat Brown published in JAMA, showed that one of the reasons that the blina arm was better was not just that it improved relapse-free survival, but it markedly reduced infectious toxicity. And that was in the US, where we put everybody on way too many drugs, probably in anti-infectious agents anyways, and we probably have less. It did that. So one could easily argue that immunotherapies are probably going to have less infectious toxicities, at least for B-ALL. I think it's important for countries like yourself, though, like we right now in the US have randomized trials right now looking at blina versus in our standard risk kids, and I know in our high risk kids, we may find that they aren't more effective in the US because infectious toxicities aren't as big of an issue. They're an issue, but they're not as big in our frontline setting. Like we don't have that many kids who die of infections in newly diagnosed B-ALL in the United States. So if we don't find a difference, I think you will have to scrutinize those data, because it could be that if you increase the infectious risk, then if you did the exact same trial in India, you actually might find that the blinatumomab arm is clearly superior because of that difference in toxicity. I think that's one of the lessons we learned on 1231 in T-ALL with Dex versus Pred. When we changed the steroid, we had way more children who died of infections. And some patients, that was offset by the benefit in decreased relapse-free survival versus others it wasn't. And I think immunotherapy is going to be the same issue, that if you add in immunotherapies, does it improve survival enough by reducing relapses versus the impact on infections enough to make a difference? And that could be something that's different in different parts of the world based on one, disease biology, which is going to be different, and two, based on the risk of infectious toxicities. So you may find, and it's very reasonable to think, you may find in India that immunotherapies are going to be even more impactful than they might be in the United States, if that makes sense. Did I answer your question?
Thank you.
Thank you. Ruchira?
Good evening, Dr. Tichy. Two questions I actually had. One was with regards to T-cell disease, where you mentioned that dasatinib may actually be very effective for treating our children who got T-cell disease. Could you identify which cohort would actually benefit from those? And the second question was regarding inotuzumab and blinatumomab. Are there any randomized controlled trials to say which of these two might actually be better? Because there's a lot of cost difference between the two, which we find in India. And simply asking for blinatumomab, giving it for a month, becomes very, very expensive for our patients.
Both great questions. The second question I'll answer first is, right now in pediatrics, there aren't randomized trials, but one of the things we're talking about in our next generation of COG trials, because this trial is about to end in pediatrics, is actually to start to ask them of those randomized questions. I think we need to see first if either one of them works. The adults in the US are starting to open those trials. There's trials that are opening up using combination immunotherapies, and there are ones that are randomizing them. So I think we may start seeing a signal there, because we have the same, in the US is the same issue too. Blinatumomab is not the easiest drug to give. It's expensive. You have to have a backpack, you have to come to clinic, you have to get it replaced, you have to, so it's just a technically tough drug to give. And there are, I think one of the questions being asked in our frontline trial in BALL is the if let's say it improves survival by 1 or 2%. So you treat everybody with BALL, low standard risk disease of blina, and it's a teeny improvement in survival. Is that worth the cost and the hassle or not? It's going to have to be a home run to do that. Your other question was on T-cell, right? What was it? Remind me again. Sorry, I'm forgetting after talking.
The dasatinib and T-cell. Can we identify which cohort to use it in?
Great question. So I think right now the data in dasatinib are compelling enough that if you had somebody with relapsed T-ALL that has an ABL class fusion, for example, which isn't common but happen, you might want to consider using dasatinib. One of the things that we're doing right now is we know if you use ex vivo modeling, so we basically take T-ALL and treat them in a petri dish, 40% are sensitive to T-ALL, but we don't know which ones that is. We know it's LCK upregulated, but there's no way to clinically do that right now. So one of the things that we're working with Jun Yang's group to do is, he's part of that big sequencing effort I mentioned. And we're applying his dasatinib signature in that small number of patients to that whole cohort of 1,350 patients that we have genomics on now, to see if we can then use that big genomic data set to identify which patients of T-ALL may benefit using technologies that are more easily accessible. So I don't have the answer for you today, but I hope to have that answer in a year. Within the next year, we're now starting, like we literally just sent his lab the RNA-seq files from the 1,350 kids. So, and he's now integrating that with his pharmacogenomic studies, with the hope that we will be able to say a year from now, this is the child with T-ALL that at least looks promising enough that they might benefit from dasatinib, that we should do a clinical trial. And the same child, if at the end of therapy, they're not responding very well, we should jump to dasatinib now. So again, don't have the answer today. It's an excellent question, but we are actively looking at that, because I think dasatinib is I like it a lot because of how easy it would be for so many people to actually just move into T-ALL quickly. We know how to safely give it on a chemotherapy backbone. Most of the drugs I talked about otherwise, ruxolitinib, mTOR inhibitors, venetoclax and navitoclax, we don't know how to give them safely on frontline ALL therapy. We know how to give dasatinib in frontline ALL therapy.
Thank you. Abhilasha?
Thank you, sir. So my question is regarding rituximab. So rituximab is beneficial for CD20 positive BALL in adults. Is there any data for the use in children and adolescents in case of CD positive pre-BALL?
It's another great question. There's no, I don't know what, I think people were more excited about the INO data and the blinded data, that those actually got to be the immunotherapies that move forward, because I think in the US, we kind of got burned by epertuzumab. So Elizabeth Raich ran a trial for the COG 20 years ago where we used a naked CD22 monoclonal antibody, added it into chemotherapy, and it wasn't effective in relapsed ALL. And because of that, I think our cooperative group soured to naked monoclonals and then jumped on the bandwagon for CARs and BiTEs and conjugated monoclonals. And then all the data started coming about rituximab in adults. So I think there's a place for rituximab in relapse and refractory BALL, but there are no ongoing studies in the US in kids that are really looking at it. There are, kind of on the same note of inotuzumab and blinatumomab randomized and combined, there are adult studies that are actually looking at integrating rituximab and blinatumomab and inotuzumab and still continuing to look at it. And we may, if those studies show a lot of promise, then we may decide to leap on that. I can say at my center, the place that we've used rituximab some is in the relapse setting in kids where the goal is to either get them to transplant or to get them to CAR T. We sometimes use rituximab as bridging therapy as part of other chemotherapy. We also use it in the multiply relapsed palliative type setting too, because it's a very well tolerated drug. So we'll have somebody with, nowadays, I'm sure you guys are seeing the same thing as us, that It used to be relapse BLL, you got one shot at it. Patient was diagnosed BLL, they relapse, you tried something, you get them a transplant, you don't, but they didn't live that long. Nowadays, BLL seems to be becoming more like neuroblastoma. They relapse, and you try dasatinib for 2 months, that gets them in remission, they do okay. Then you try inotuzumab, that gets them in remission for 2 months, they do okay. Then you do a CAR, you prolong things. I think when I gave a talk here almost a year ago, there was a case you guys presented, which was very similar. Like you had this patient who had fungal disease in the brain that you kept alive for 3-4 years by using all these new therapies that were not very toxic to just continue things going. And I think that's what BLL is becoming. And I think that's where rituximab is one way that it fits in, is in this whole give the patient good quality of life to continue something that's not going to probably cure them, but may at least help. Again, in the US, rituximab doesn't have much legs, though, I think, in pediatrics in the frontline setting, for all the reasons I just mentioned.
Thank you, sir.
Thank you. Harsha?
Thank you, Dr. Tichy, for wonderful presentation. My question is with regards to BCL ALL, persistent MRD positivity post-consolidation. Is there any option of chemoimmunotherapy alone without transplant? If so, which group of patients you would choose?
Great question. A little bit of an unknown. So right now in the COG, we have this study where we're taking patients who are N-consolidation MRD positive BLL and just treating with CAR T cells to see what happens. The hard part is it's not a randomized study. An ideal study, we would have randomized those patients to transplant versus CAR alone, or CAR followed by transplant, because it's going to answer the question of how active it is, but it isn't going to answer the question of how curative it is. I think that patients who aren't on studies, the standard of care for patients who are N-consolidation MRD positive is still transplant. So I think that you use things like immunotherapies or CAR to get them to transplant. And maybe someday we'll learn that's not the best thing, that CAR on its own is the best therapy, or you can use immunotherapies to wipe out that last bit of MRD and then put them back on chemo. I don't think we know it enough now. What we know is that transplant does salvage a lot of those patients, and it's better than anything else we have. So my personal bias is we should still be transplanting those patients and using our novel agents as a way to get them to transplant. There are people, though, right now that I know across the globe, and people in my own center who disagree, who think that you could use like a CAR in this setting as definitive therapy. I don't think we're there yet.
Thank you.
Thank you. Adish?
Thank you very much, Dr. Tichy, for that wonderful presentation. I have one question regarding a B myeloid mixed phenotypic acute leukemia. So if the B myeloid leukemia is positive for BCR-ABL, and we go about MRD, doing MRD post-induction and consolidation. So what do you suggest, BCR-ABL quantitative MRD or flow cytometry based, or we should do both? Because in India, we are a cost constrained country. So like I have gone through some papers where they suggested that both should be done. So what is your idea about MRD positivity and MRD assessment? Whether quantitative PCR, BCR-ABL should be continued, or we should do flow cytometry based MRD also?
So I'm a little bit biased because the US, where the cost isn't as much of a thing, we're still doing both. So it's hard to really give definitive guidance on should you be doing one versus the other. I think if I had to choose in BCR-ABL and you can't do both, then I would err on doing the molecular testing. I think it's better. But I think there is a little bit of discordance, and I think the problem is in something like B myeloid BCR-ABL, which is very rare. The question is, what is the threshold that you use to transplant the patient? And I think one of the reasons in that rare group that you might want to do both is because I think if you had somebody who, based on the amount of response, I mean, most patients with mixed lineage, biphenotypic, ambiguous lineage, whatever you want to call it, ALL, a lot of them need a transplant. And some of them need myeloid therapy, and some of them need targeted therapy. So for me, the more info you can get, the better, to try to make those big picture decisions on how well your patient is doing. At the same point, you don't want to transplant something who doesn't need a transplant. I think the other thing we need to do in these populations is ask more questions. Like, for the first time ever, in the COG, we're actually allowing patients with biphenotypic leukemia to enroll in our trials. Like that was always been an exclusion criteria. So we have no data at all. It's all been retrospective, 10 patients from this center, 20 patients from this center, pool them all together, see what they got and try to figure out what's the best. And that's a horrible way to like learn the best approach. So I hope one thing that comes out of our current trial that's basically not asking a question other than taking everybody with mixed lineage leukemia, putting them on the same backbone, treating them the same way, assessing MRD the same way, deciding on AML therapy the same way, deciding on transplant the same way. And then at the end of the day, just have a cohort of patients that you can look at and try to understand how do they do the same therapy to then design future trials is probably an important step.
Thank you.
I wish I could give you a more definitive, but it's such a hard, the biphenotypics are so hard because there's just so little data on what's the best way to go. And we also lump them. Like we take patients with B myeloid and T myeloid and treat them the same, which is probably, we don't do that with B-ALL and T-ALL. Why do I think we do that in B myeloid and T myeloid disease? It comes up, should we be giving nelarabine to our T myeloid kids? Oh, we don't have any data.
Archana.
Yeah, first of all, thank you, Dr. Tichy, for the very, very interesting talk. So my question to you is, with conventional treatment, we see a lot of toxicity in the Downs babies, and also some kids just totally don't tolerate the chemo agents. What would your plan be, given the new agents, to treat these children? Maybe not now, maybe sometime in the future.
So
how would you draw out a protocol or a plan for the treatment?
So AJ Vora and the MRC presented data at ASH this year on the use of blinatumomab in infants in Downs and infants both. They actually looked at it in some of that, and it's looking very promising. In the COG, we're doing a study right now for Downs, not infants, but just kids with trisomy 21, where we're actually replacing cytotoxics in some blocks with blinatumomab to try to reduce toxicity for those issues. So I think the answer is there are ongoing efforts that are looking at these questions, both in Downs and in infants. Blina is the main focus of those right now, but the early signals are looking pretty promising. So I think we're going to start seeing those data over the next year or two. At my center, for both groups, we continue to treat with cytotoxics, but we have a low threshold to move them to an immunotherapy-based approach if they're having a lot of toxicities or poor responders or having other issues, especially the babies. Like, so for the infants with ALL, we are quickly moving them to immunotherapy-based regimens off a clinical trial if they're not doing perfect with current therapy. Where we're still using our goal is to get them to transplant. I think one of the nice things about immunotherapy in the infants is you can maybe string them along longer so they can at least get to be older, so their brain's a little bit more mature. When you do take them to transplant, you may not have the same morbidity. Because the problem is most of our infants, when we do take them to transplant, we do a non-TBI based transplant, and that's just not as good for ALL. So I'm kind of hedging a little bit because I don't know the answer right now, but I think in the next couple of years, we're going to learn the answer because there's a lot of exciting stuff, a lot of it coming out of Europe that are looking at those questions.
Chetan.
As always, Dr. Chi, excellent overview. Thanks a lot for that. My question is on two things. The first question is on ponatinib and blinatumomab in the PH positive ALLs. The adult data looks pretty promising. Anything on those lines in PH-like or PH in pediatrics coming up?
So the next PH-like study that's in development at the COG, we'll see if it happens, is actually to look at adding an immunotherapy in that population. So to continue moving forward with TKIs, but then randomize to receive or not receive blinatumomab and immunotherapy. And that's going to be a joint US and European trial. I say the plan for that is because it's still early in development. People are very enthusiastic about it. Titron with Mignon Low, we're developing that trial with Ceratagen, and it could undergo 1,000,001 alterations before it opens, because the problem with PH-like ALL is it's rare enough that, and outcomes are decent enough that in the US, we can't do a randomized trial. We just don't have enough patients. It would take us 20 years to do it. So we're trying to partner with Europe right now to see if we can improve our patient numbers, but it is complex doing internet. We need to do more international trials, but because of the regulatory issues, it is highly complex to do a trial both in North America and other countries currently. The FDA puts restrictions on us. The NCI is very particular on how they want data captured. They insist that they want the data captured the same at all sites. So while in the US, we have the same systems in the US for capturing data on our clinical trials, the Europeans have different systems. I'm sure you guys have different systems for capturing data on your trials. And the US government mandates that we use the same system. So we don't want to change ours, they don't want to change theirs. It just gets to be more complicated. So another hedge a little bit, I think that probably is where PH-Lite needs to go, which is ask questions of TKIs plus immunotherapies. And there are efforts to try to do it in kids, but they're still in development.
Thank you, Chetan. Are there other questions from faculty here? We'll take up some. Okay, we'll take up
Sir, can I ask one more?
Yeah, of course.
So I was really interested in your daratumumab data, even from the last talk. I was very impressed with it. I just wanted to know, with the use of daratumumab, how does the patient tolerate it? Because the immune system will collapse with the T cells going down, the NK cells and the B cells, all of them will be affected. So is it just a bridge to transplant?
Great question. So right now we've been using in relapsed TLL. I still believe that transplant is the goal. And actually one of the endpoints on that trial that we're most happy about is, well, if I can't say the specifics, the number of patients who are on the early phase trial who made it to transplant. Thus far, the safety signal is very good. Patients tolerated daratumumab added to cytotoxic chemotherapy very well. We had on that trial, which had like 30 patients, there was one toxic death, and that was not daratumumab related, that was asparaginase related. And then there were some other, there were patients who did have toxicities from all the nasty cytotoxics we have, but no more than were expected based on just using the chemo backbone alone.
Great. Thank you so much.
Thank you.
Looking forward to some of that data.
It'll be out soon. Finally, the trial's done, and they're getting ready to submit for presentation and then hopefully publication this summer.
Okay. Any other question? Yes, Shripat?
Yeah, one area which I really feel, we have not done much progress over the years is the hypodiploid ALL, So any thoughts, anything new, like one of the agents that we use in relapse setting, like the mitoxantrone, or even the newer drug like HDAC inhibitors or something, you think there will be other drugs which will be important in the treatment of hypodiploid ALL, or the maintenance that we use now is not the ideal maintenance for hypodiploid and we need to change that?
So there's preclinical data with hypodiploid ALL from Charles Mulligan and Mignon Low that looked at it genetically, and then in preclinical models that the venetoclax actually looks pretty promising in preclinical models of hypodiploid ALL. And I think there's talks about, could we develop a trial to test that? It's hard. Hypodiploid's hard to do trials in because it's so rare that it makes it hard to really do a study. But one could envision that if you had a venetoclax trial in relapse refractory ALL, like T-ALL, that you could maybe have a hypodiploid arm to at least get some data on that. And then, of course, I think the other thing people are hoping that'll be promising are immunotherapies for hypodiploid. I think there are enough immunotherapy trials that are out there that are now starting to include hypodiploid patients. I think we'll learn those outcomes. The outcomes are so bad, it might be, even though they're non-randomized, we might be able to see enough of a signal. I think it's unfortunate the hypodiploid, the frontline Tisagen Lec-Lucel trial we did in COG, we're basically taking patients who are refractory disease and putting them on CAR T cells in first remission. That actually excludes hypodiploid patients because all of the data we had on in-consolidation MRD, historical data, didn't include hypodiploid because those patients were taken off therapy and allocated to transplant. So we didn't have chemotherapy-only outcomes on them as the control group. So I think that's one unfortunate thing, but I think we're going to start hopefully being able to ask some questions in that group. And I think to answer your question, at least the preclinical data I've seen, BCL2 inhibition and immunotherapy are probably the two most promising things out there.
Thank you.
Thank you, Sripath. Any other? So can we take some question from the audience? Dr. Tichi, can we take some questions?
Yeah, sure.
There's a question. What should be the cutoff for CD22 positivity for using inotuzumab? 20%, 50%?
What was it's a great question. I don't think there's good data that actually say what the definitive cutoff should be. I think on our current COG trial that's randomizing patients in the front line, you have to be positive at some degree, a low level degree to get enrolled, but it's very low. And I think we're going to start asking those questions as the trial data mature to actually say who's more likely to benefit, those that are 50% positive, 20% positive. It gets so complicated at 22 though, because the question is it, you want 50% of your blast to be CD22 positive, or is it antigen density that matters? Like, so if you have 100% positivity, but it's low density, is that worse than having subset positivity? I don't know the answer to that. I think it's a great question. We're looking at it, but I can tell you at my center right now for inotuzumab off trial, we're usually making the decision. We're almost doing it as a yes, no. If you have positivity, you go. If you can detect it, if it's negative, we don't try it. But we're not getting nuanced. We don't feel like we know enough yet to be nuanced enough about a cutoff.
Right. BCL positive acute lymphoblastic leukemia, molecular test becoming negative, flow becoming negative. Does he require upfront transplant?
We're not transplanting those patients.
And how long do you continue the TKI?
Currently, we're following our COG chemotherapy backbones, and we follow the protocol, which for the pH positive patients, we usually do it through maintenance and then stop.
Thank you. Any guidelines for treatment of ETP ALL in community practice?
So right now, I think most of the data from across cooperative groups suggests that ETP ALL has the same outcomes as regular T-ALL, and we are treating them the same. I mentioned there may be the data from 1231 that may show that ETPs did worse on a DEX-based backbone. That may make us rethink that, but we haven't really dived. It's a new thing we just saw. We haven't dived into them in depth. So my ultimate answer is ETP and non-ETP TLLs, treat them the same. Same MRD cutoffs for transplant, same chemotherapy backbone, same everything.
Thank you. The question is there importance of vincristine steroid pulse therapy during maintenance?
I wish we could get rid of it. I think a lot of the world has. We still do it in the US because we haven't been able to get rid of it. I really think it's pointless in T-ALL in particular, where maintenance doesn't probably really matter. In B-ALL, my guess is it doesn't help. But at least in the US, we still use it as our standard. We're trying to decide if we can do more trials where we try to get rid of them. It's hard to convince people to non-randomly eliminate something in the US. And the problem is it's a tough study question to ask because it's a non-inferiority design. So to say no pulses in maintenance versus pulses in maintenance, you have to say, is it the same? And non-inferiority needs a lot of patience, and people are nervous to make the leap. And then the question is, do we spend 8 years asking a trial to get rid of the pulses? And then people don't get very excited about doing that. So we want to get rid of them in the US, but we haven't been able to. My personal bias is they don't do anything.
The question is there an age cutoff for using the pediatric protocols of treating ALL?
That's a good question too. I think that the adults are using them older and older now. I know that, so recently in the US, there was a push by the NCI to do something called the Alliance Initiative, which is basically to say for adolescent young adults to force people to treat them on pediatric or ALL trials to at least treat them the same way. And through doing that in ALL, our adult colleagues are now treating adults up to age 40 easily with pediatric protocols. So I think for T-ALL in particular, they're now, the standard in the US is up to age 40. They're now treating with 0434-like therapy. There's now centers in the US that are treating adults 40 to 50 with pediatric protocols. And there are people that are older. I think the one drug that people have to tailor differently in adults versus kids is asparaginase. So adults just don't tolerate asparaginase as well as kids do. So while a lot of my adult colleagues are now using pediatric protocols or protocols that are very similar to pediatrics, they do reduce the asparaginase, they cap the dose, they adjust it more based on obesity and BMI, they use less doses of it. So I think that's the one place that you need to, it's pediatric therapy light.
So there are a couple of questions about asparaginase. The European dose of asparaginase is much less than the one used in US. Is the US dose required?
Well, I think part of that's formulation too. So we don't use exact same formulations. And I think some of the dosing is different. We do push the dose higher in the US than other groups do. I think I'm a big believer that you should you should pick a backbone and a regimen and follow it. Because the outcomes are actually pretty simple. Like if you look at the outcomes on European regimens versus US regimens in B and T-ALL, there are differences, asparaginase dose, maintenance pulses, number of ITs, that kind of stuff. But at the end of the day, the outcomes are the same. So I would say pick a package. So, and don't like say we're going to use the US asparaginase dose and then use the European number of ITs. I just think that you should, I'm a big believer in the outcomes were 90% using this regimen. So pick it, and then it doesn't matter which one it is, but pick one and stick to it.
Another question is, can asparaginase be given with TKIs for pH positive ALL?
Yes.
Okay. If there is a patient who has anaphylaxis to asparaginase, you just drop it or change over the protocol to something like hyper CVAD?
We in the US typically just drop it and continue, knowing the outcome will drop a little bit. It's not enough that we think that it's worse. We still think our outcomes are better than like hyper CVAD. So we just continue doing the same without that one drug.
Right. I think rest of the questions, most of them you already answered. So one or two questions about how do you decide between the two types of immunotherapy, inotuzumab versus blinatumomab, with respect to extramedullary relapse, with respect to CNS relapse?
So CNS relapse, we typically we favor blina more because there's more data on that, honestly. For us also, the other big thing is what's the goal? Is the goal to, and sometimes you don't know, is the goal to take him to transplant? When is it to take it to transplant? How worried about VOD in this patient based on what they've seen before? And then I know has that extra VOD risk. Do we think they're probably are they a high enough risk patient that they're probably gonna need CAR T cells? Is the goal actually just to bridge them to CAR T cells? Is it a goal to bridge them to CAR T cells and transplant? And then it gets nuanced in there because we actually, for kids off study, we take all those things into combination to try to decide what is our ultimate goal. I mean, there was data that were just presented published in JCO this past month that actually looked at like, what's the impact of having blinatumomab on response to CAR T cells? And they basically said that there's a group of patients who don't respond to blinatumomab, and those patients probably don't respond to CAR, but the ones who do need a transplant because they're going to relapse. So I think there's more we're learning now about the impact of different immunotherapies on other immunotherapies and transplant, but I don't think we know enough now to just pick. So I'll be honest, at our center, it's patient by patient still, where the ultimate thing is, what we always discuss is what's the goal? Is the goal transplant? Is the goal CAR T? Is the goal chemo only? And then that informs what toxicities they had, and that kind of informs which one we pick. Is it hard to give them blinatumomab? So is this a patient that's relapsed, that the goal is not transplant because we don't think they're going to survive it? And then what's better quality of life for blind is a pain?
Chetan, you have a question?
Yes.
Yeah.
Dr. G, you mentioned about VOD after inotuzumab in transplant. So, any particular toxicities in transplant post blinatumomab? 2 cycles?
Not that I've seen. I haven't seen anything in the blind literature that says that they do worse with transplant than chemo. If anything, it's better from what I've seen compared to cytotoxics, because they have less infections going into it, so they have less infectious mortality post-transplant.
Thank you. Another question is for T-cell ALL. Based on your lecture, can we reduce the duration of maintenance therapy for T-cell ALL?
So as in my center and in a lot of US centers, we already have non-randomly. At least we treat boys and girls the same. So we've gotten rid of that last year for boys. The next trial will do the same thing non-randomly. But beyond reducing it, beyond the 2 years, which is like 2.5 years of total therapy, I think we'd have to do a trial for that, because right now our therapy in TLL is curing a lot. We know that in TLL, 2/3 of relapses happen early. So they happen before you get to like the second month of maintenance, which is why I say maintenance doesn't matter as much in TLL. On the flip side, 1/3 of patients do relapse in maintenance. So there's some benefit to maintenance, but I think the relative benefit to maintenance in TLL is far less than BALL, but I do think there's some benefit to it.
The question is, if the MRD is persistently negative, do you avoid bone marrow transplant in otherwise genetically a bad ALL?
Good question. Depends on the bad ALL. 17, 19, yes. We still take all those kids to transplant. Hypodiploid ALL that gets MRD negative at my center, we don't transplant them. We do chemo alone, mainly because we know the outcome's still not great, but transplant doesn't make it better.
Right.
I see one question that's popping up on my screen about, we don't do anything differently based on IKZF1 mutations currently, or deletions.
Yeah, I think the rest of the questions have been answered. So if there's nothing from the faculty here, I thank you on behalf of Mumbai Hematology Group, and for the official vote of thanks, we have Amcure. Keyur.
Thank you so much for giving this opportunity to associate with Mumbai Hematology Group and Pediatric ALL webinar. Thank you, Dr. David, for sharing your expertise. And thank you, Dr. Agrawal, for giving us this chance. So we are solely working in India for pediatric ALL treatment, starting from the Hamcell, means which is PEG asparaginase, till we are working on Erwinia, which will be developed in next 3 years' time in India. So we have, Dr. Banawali has already developed the molecule in terms of pediatric PEG asparaginase in Tata by doing the PKPD trial. And rest of the molecules also coming up in India very soon by Amcure, sir. Thank you so much for giving us the time, sir. Thank you so much.
Thank you very much for inviting me. It was my pleasure. Hopefully one of these days, the pandemic will get to be a point that I can come out there and meet you guys in person.
Sure. Thank you so much, Dr. Tichy, for sparing your valuable time for us.
Thank you. Bye.